In this video, we're going to look over and review the different levels of structure for a protein. So in Module 4, we already discussed primary structure, and so that's the amino acid sequence of the protein. So that's the unique sequence of a particular protein. And so the main thing we're concerned there, of course, is the covalent linkages or the peptide bonds between the amino acid residues that make up the protein. So you might think of the dominant bond that's associated with primary structure as a covalent linkage. Okay. So moving up though, because what the protein eventually does is this primary structure dictates what the three-dimensional structure will be of the protein, right? So the next layer or the next step up in structure is known as secondary structure. Okay, so there are motifs that are commonly associated with secondary structure. So that includes an alpha helix, such as that's shown here. There are also beta sheets, which we'll discuss more, and other motifs such as beta turns. Okay, and so the primary intermolecular. So now we're looking at intermolecular interactions that are sort of dictating these structural motifs, and the primary intermolecular interaction, or weak interaction, I should say, that we're looking at is hydrogen bonding. And that's hydrogen bonding that we're considering with the backbone. We're not even considering the side chains at this point. Okay, so these are H bonds between the carbonyl oxygens of the peptide bond, and then the NH hydrogens also of the peptide bond. And that helps to stabilize these secondary structures. From there, the secondary structure, once secondary structures are considered, this helps determine what the tertiary structure is. So this is what we think of as a three-dimensional structure for a protein. Okay, and tertiary structure is going to be dictated by a number of factors, the most significant of which is going to be the hydrophobic effect, because when these secondary structures develop, they can often have regions of nonpolar and polar amino acid residues. And so what's going to happen is that the nonpolar residues, regions with nonpolar residues, will collapse on each other in order to avoid interaction with water. And so this hydrophobic effect is largely the determining factor of tertiary structure, but there are also other enthalpic contributions, such as salt bridges, which are electrostatic interactions, hydrogen bonding, and of course other weak interactions, including van der Waals forces that help to dictate what the tertiary structure will be. Finally, quaternary structure is a fourth level structure that only applies if there is more than one polypeptide involved. So the peptide itself will fold into some tertiary structure, and then this may interact with a second polypeptide that also has its own tertiary structure. And so quaternary structure is only considered when more than one polypeptide is interacting. So one example is hemoglobin, which has 4 subunits, 1, 2, 3, 4. So those are 4 different polypeptides that interact with each other to ultimately make the protein hemoglobin.